take a quick look at how you can use NCBI to check on your databases. So if you start from the beginning. When you start with your home page, you can put in NCBI in here. NCBI, and it will take you to this. When you get there, you've got a few options. One, you can put in your organism and just click all databases and search, or you can go directly to the genomes and maps part of the database and check what's there. So if we just take a quick look to see what's here, these are all the things in that section of the database. And it also has more information, things you can download, tools you can use to learn, and how to do stuff. So this might be a bit much for you. So all you're going to do is just keep it at all databases and then put in your organism, search. When you do this, what will happen is it will tell you how many databases it's got hits on and how many genomes. So if you look at this, these are all the genomes for your organism, and this one with the green tick is your reference genome. So this is the one that is used as the standard. These are all the other times other people have sequenced this particular organism. All these are different strains, but it's the same species, right? And that's all the data you have. What can you do with this? You can actually look at the actual assembly, or you can go over here and view the details, view the genes, or download it. So you could download it and actually work with it on your device. If we view the genes, you're talking about wanting to annotate this genome. This is how it is already. What's an annotation? Look at this. So here we can see that the orientation of this particular gene, the gene symbol is this, the gene name is that, the orientation is going forward on the main strand, whereas others will go in the other direction. You remember we talked about annotations. So basically, there's all this annotation data. This is the length. Like if we took one particular one, let's take this one. Aspartokinase 1. That's the symbol. What it does, it's a protein coding gene. The length, how many amino acids? 820. So this is already annotated. But there's some which are putative. Here we are. So this is a putative cytoplasmic protein. It goes in the other direction on the strand. This is the symbol they've given it in the annotation. And it's a protein coding gene. And it's 257 amino acids. It's putative because you don't know what it does yet. The ones that people have tested in the wet lab and they know what they do are just written like that. This one's a threonine synthase. This one is putative because nobody has actually established whether this predicted protein does whatever is said in the prediction. So if you look through here, all these are putative. This is a putative protein. This is a putative protein. This one's hypothetical, meaning that it's not even the threshold for it to be named a protein is a bit low, so it's hypothetical. It might be a protein, it might not, we don't know. So all these are the ones that you could, in the wet lab, go and investigate to prove what they are. Right? So where you wanted to do the annotation of these genomes, this has already been done. What you could do is think of proving whether one of these putative genes is actually what it's claimed to be, like this one. Is this really an exochitinase? So you could think about how you'd prove that this is an exochitinase. That's actual work to do. So that's why you have the wet lab and you have the bioinformatic people. They work hand in hand to give us predictions and actual checking to see what the prediction made, whether it's true or not. So there's better things we could do with these databases, and that's what your project should really be about. How do we use this data? This has already been done.